My name is Ola Hössjer. I live in Sweden, a suburb of Stockholm. I'm 60 years old. I have 2 daughters and 1 son-in-law. And I'm a member of a Stockholm Vineyard Church in Stockholm. And I work as a university professor in mathematical statistics at Stockholm University. But I'm also part of the Swedish creationist organization Genesis, and I want to send my warmest greetings from them. And the topic of this talk today is an overview of Baraminology, the systematic scientific study of biblical kinds. So we read in the first chapter of Genesis that God created animals and plants after their kinds. And it turns out that this is a very reliable description, and science can confirm this. And what I'm going to say today, this talk is based on one chapter from this book. Last year, in October...
You don't need this microphone.
Oh, yes, you're right. Yeah, thank you. Last year, we had the first Nordic Research Creation Conference in Malmö on this topic, the origin of diversity in biological life. And the speakers of that conference, they agreed to write 1 chapter each of the proceedings that will come out in a couple of months, I hope. And Tommy Alto here is one. He has written 2 excellent chapters on epigenetics. Boris Schmittgall, the CEO of Wort Wissen, has written another chapter on the origin of life, and I will speak on biblical kinds. So let me give an outline of this talk. So I will start mention about the bushes of life model, and that is really another name for God creating animals and plants after their kinds. And then I will turn on and give a brief history of the systematic study of created kinds. And that's where the word baraminology comes from. It's a Hebrew word. I will come back to that. And then you could say that this systematic study has 3 major goals. And the first goal is to really find those kinds. Which are the created kinds? Which groups of animals do they correspond to today? And then the second goal is to find the mechanisms of divergence and speciation within kind. So for each group, each kind, it started with a single pair or a first generation, but today we have many species within each kind, and it's like a bush. But the different bushes, the different kinds, they don't have common ancestry. So this speciation or adaptation to environmental changes, that is not evolution because it doesn't create new structures. And then we have the third goal of baraminology. Given that you found those created kinds, trying to find the ancestral history of each kind. And then we will zoom in to humans and ask the question, is it a kind of its own? So do we humans have unique ancestry from Adam and Eve that were created uniquely in the image of God without ancestry with animals, as the Bible tells? And then I will wrap up with some conclusions. So let us start with the bushes of life model. So you could roughly talk about 3 models for the diversity of life. The classical creationist old model is a species fixity model, that God created species ready 6,000 years ago, and then they haven't really changed. And then we have the bushes of life model, which I will talk about today. It's also creationist, but it says that God created the first pair within each kind. And then there has been speciation within each kind, but the different bushes, the different kinds, they don't have common ancestry. And then we have the evolutionary tree of life model, where all living species today, they all descend from first unicellular organisms. So they all have a common ancestry, and that gives us the tree of life model, usually with a much longer time frame. Instead of 6,000 years ago, several billion years. And you could say that the bushes of life model is in between the species of fixity and tree of life model. Although the bushes of life model is creationist, it does allow for speciation. But as I said, it's another type of speciation that evolutionary biology holds, because evolutionary biology, the tree of life model, requires that new species with completely new types of organs, wings and eyes and echolocation systems and so on, evolve. So let me give some examples of kinds and bushes. So although strictly biblically humans are not a kind, because we are created uniquely of our own, but we can also think of humans as our own kind. So humans, modern humans, Homo sapiens, but then also include Neanderthals, Denisovans, Homo erectus, then they all have a common ancestry from Adam and Eve. So we can say that they form their own kinds. But then we also have great apes, chimpanzees, gorillas, orangutans, they form their own kind, Pongidae. And then we have the dog kind, Canidae, consisting of wolves, foxes, breeding dogs, and so on. We have the cat kind, Felidae, including tigers, lions, jaguars, cats, and so on. And so these are 4 examples of kinds. But let us now go back to the Bible and ask ourselves, is there a biblical motivation for kinds in general? And first of all, as I mentioned earlier, we go to the first chapter of the Bible, Genesis chapter 1, and in several verses, we are told that plants and animals were created after their kind. And then at the end of the first chapter, and then the creation story continues in the second chapter of Genesis, telling us that humans were created uniquely. So we can think of humans as its own kind. But then we have diversification, we have speciation within each kind. And we can also think of a biblical motivation for that, because not only humans, but also animals were commanded to multiply and fill the earth. And we can think of God designing mechanisms of environmental adaptation, including speciation of animals within kinds. And if I'm allowed to speculate a little bit, we know that God can look into the future. He knew about the fall of man, and he knew that also the nature will start to decay after the fall of man, and that would create some harsh environmental conditions. So maybe God created these mechanisms of adaptation, also having those harsh environmental changes in mind from early on. But then, of course, we have a limiting factor, the flood of Noah, 1,500 years later, 4,500 years later, which also relates to the title of this German book that I mentioned about. So you could phrase this as a bottleneck. So the size of each animal kind was reduced to 2 or 14. So in some way they had to restart those kinds after the flood of Noah. So that was a biblical motivation for the bushes of life model. And let's now relate this to Baraminology, which is the title of this talk. And Baramin is a Hebrew word for created kind. It's really composed of 2 Hebrew words put together. And Baraminology is a systematic study or systematic scientific study of created kind. And the word was introduced by the creationist Frank Marsh in 1944. And as I mentioned, when I gave the outline of this talk, there are three major goals of Baraminology. First of all, to identify all created kinds, which are they, and then find which are the mechanisms that give this speciation and divergence within each created kind. And then thirdly, given that we have found the created kind, find their ancestral history. Which individuals or which species within each kind are more closely related to each other. And in order to study this, we will actually be helped by taxonomy. And both secular sciences that hold on to the tree of life model and creationists are helped by taxonomy, because in taxonomy you sort life hierarchically into levels. We have species at the next level, the genus, at the next hierarchical level, higher up, family. We have the order, we have the class, phyla, kingdom, and domain. And an evolutionary biologist think that all species are related through the tree of life, but as a creationist, you believe that species are only related within the same bush. But we can still have the taxonomic levels can still help you, because the lower orders of taxonomy can correspond to species within a bush. And then, of course, we can have common design between bushes, so that some bushes can be more related than others, because God, as a designer, can reuse the same principles for different bushes. And that leads us to the question, which is the typical taxonomic level of a bush kind? Is it at the species level, the genus level, the family level, or the order level? And Erik Wassmann, more than 100 years ago, a creationist, he said that, well, it actually depends on the study group. And 15 years later, George McCready Price, another creationist, he said that, yes, but family is often a good candidate for being the right taxonomic level for a biblical kind. And it turns out that both of these predictions were good. So let's suggest some possible kind levels. And I will partly go back to the examples I gave earlier of biblical kinds. We have humans, as I said, including modern humans, Neanderthals, Denisovans, and we think of all of them as descendants of Adam and Eve. And then humans form its own kind at the species level. On the other hand, the other three suggested kinds that I mentioned, great apes, the dog kind, and the cat kind, taxonomically they are all defined on the family level. Whereas many creationist researchers think that chicken-like birds, galliforms, they form its own kind, and it's yet another hierarchical level higher up at the order level. So the hierarchical level of a kind might depend on the study group. So let's now turn to the history of the bushes of life model and its alternatives. And up till the 18th century, young earth creationism was very mainstream, so most Christians were creationists, and the species fixity model dominated. But my countryman, the Swedish botanist Carl Linnaeus, the father of taxonomy, he also advocated species fixity. But at the end of his life, he actually changed his mind and allowed for some speciation within, actually. But he was still a creationist. So that means that already in the 18th century, he opened up for the bushes of life model. But then we know that at the end of the 18th century and in the early 19th century, many scientists adopted high H's after the work of geologists, the Scottish geologists James Hutton and Charles Lyell. And that in turn gave rise to tree of life models because you needed long time frames for evolutionary model. The French scientist Lamarck, He proposed an evolutionary theory in the early 19th century. And then about at the same time, Alfred Wallace and Charles Darwin in the late 1850s. But even at this time, there were many scholars who criticized the Tree of Life model, and they favored various versions of the Bushes of Life model. For instance, the British botanist William Herbert in the 1820s, and also the paleontologist Richard Owen in the 1850s, and he was a critic of Darwin's theory. But in spite of this, that there were many critics of evolutionary theory who favored some versions of the Bushes of Life model, in his famous book, The Origin of Species, Charles Darwin, he pretty much equated creationism with species fixity. And there were so many signs of speciation taking place. So that sort of gave a negative flavor of the creationist camp. So let's continue now to the 20th century. And then we know that the Tree of Life model, it gained wide acceptance in the 1940s, because then evolutionary theory was married with genetics and giving genetic explanations for the occurrence of new species through macroevolution. And that's called the modern synthesis or neo-Darwinisms. And instrumental persons in this process were the geologist George Gaylord Simpson, the biologist Julian Huxley and Ernst Mayr, and the geneticist Theodosius Dobzhansky. But there were still many creationists around during the first half of the 20th century, and increasingly many of them favored the Bushes of Life model rather than the species fixity model. For instance, Eric Wassmann, that I mentioned about, Harold Morton, George Macready Price, that I mentioned about, Dudley Whitney, Harold Clark, and Byron Nelson. And then, as I mentioned, in the 1940s, Frank Marsh, he coined the term baraminology for the systematic study of created kinds. And I would say that from the 1950s, species fixity became a minority position among creationists in favor of the Bushes of Life model, because there were so many examples of speciation taking place. So that was some brief history of the Bushes of Life models and alternatives. So let's go back now to the three major goals of baraminology. First of all, to identify all created kinds. Which are they? Secondly, find mechanisms of divergence and speciations within each kind. How does it work when new species form within each kind? And thirdly, to find the ancestral history of each created kind. Which species within each kind are more closely related than others? So let's turn now to the first goal of baraminology, to identify the created kinds. And there are actually three main methods for doing that. The first one is so-called hybridization, or looking at hybridization observations or experiments. For which species within a kind is it possible for them to have offspring? And that signifies that they belong to the same kind. The cognitive method, that we can use human senses to help us identifying biblical kinds. And then something called statistical baraminology, where you use statistical methods for very large data sets in order to group species into kinds. Let's start with hybridizations, and suppose that two organisms or individuals, that they can hybridize and have offspring. And reproduction, that's an extremely complicated process. Here we have an example of embryological development from the first fertilized cell. In order for this embryological development to take place, the two organisms, they must be related in order to have viable offspring. For this reason, Frank Marsh in 1944, he said that, yes, hybridization is a sufficient condition for a pair to belong to the same created kind. It's not a necessary condition, because a pair of species, even though they have common ancestry within their bush, they can lose the ability to have offspring. But if they belong to the same species, Leitner et al. In an article from 2011, they said that yes, then more is required, because then the pair also needs to have fertile offspring. We have, for instance, donkeys and horses. They can have offspring, mules, but they are not fertile, but they still belong to the same kind. So they hybridize, but they don't have fertile offspring, because they don't belong to the same species. So can we define hybridization within kinds more accurately? Well, as I said, in order for the pair to belong to the same kind, the offspring doesn't need to be fertile. And it doesn't even need to survive embryological development and be born alive, but it needs to pass a certain stage of this embryological development. And that was suggested by Siegfried Scherer, your countryman, in 1993. And then you could use hybridization data between different pairs of species in order to build up different groups and thereby suggest different created kinds. So let's look more closely into the cat family and hybridization within the cat family. So as I mentioned, the cat family, it's a taxonomically it's a family. It consists of 40 species and it has 14 genera. So genus is a level between species and family. And here we have hybridization data for 6 genera. We see they are notated as G1, G2, and so forth. So we have different species, lion, tiger, leopard, and so on. And if there is a successful hybridization, it's marked with a cross. So we see that several of these different species, they can hybridize. And probably this data is incomplete, but even for this incomplete data, we can see that we have 2 hybridization chains. So at least we have 2 groups within the cat family: lion, tigers, leopard, pumas, and ocelots forming one group, and the other group. But it's still generally believed that this whole group forms its own kind. So that's the first method for identifying kinds, hybridization. The second method is the so-called cognitum method. So, and the general idea, going back from a paper by Saunders and Wise from 2003, is that it is possible to use human senses and sort species into groups. And those groups need not be at the kind level, because we can think of, yes, all the mammals, which taxonomically is higher up, they have certain shared features, or we can also look at species within a kind, like all foxes. They share certain features, and we can use human senses in order to do that grouping. And in doing so, that it's a useful complement to hybridization. If the hybridization experiments are not really complete, we can use human senses to sort of suggest that, yes, this is really a biblical kind. And let us give a biblical motivation for using human senses to sort species into kinds. So we read from Genesis chapter 2 that Adam was in fact able to name all animals. So and since then all animals corresponded to different kinds. We had not yet had any speciation. He was able to identify kinds. And similarly, Noah was able to get animals of each kind into the ark. So that suggests that he was able to recognize them. And that also, since we are descendants of Noah, we probably can do this as well. And then, so let's apply the cognitum method to the cat family. So we see here from different members of that family, the cat, the jungle cat, the bobcat, the cougar, leopard, and tiger, they do have strong visual resemblance. And we already know from the hybridization experiments that they seem to belong to the same kind. And then the visual resemblance supports the conclusion of the hybridization experiment that Felidae is in fact a kind. But apart from hybridization and the cognitive method, there's a more modern method, statistical baraminology, and in order to identify kinds. And it's used for large, either morphological data sets having to do with the forms and shapes of organs, or molecular data sets. And these data sets are so large and of the kind that you cannot analyze them using human senses alone. So we need algorithms and statistical methods to do that. And let me explain basically how statistical baraminology works. So it has three major steps. So first you need to find a long list of characters. In this case I only have 10 characters, and they could be morphological or genetic. And then for each species you list whether that particular character is present or not. So the filled circle represents characters that are present for each species, and unfilled circles means that they are not present. And then in the second step, you compute a distance between each pair of species based on the fractions of characters that differ. So we see here that the distance between species 1 and 2 is 0.1, and that is because it's only 1 out of 10% of the characters that differs. Character 6 is present in species 1, but it's absent in species 2. Whereas if we compare species 1 and 3, the distance is 0.9, because they differ in all characters except character 6, where that particular character is present both in species 1 and 3. So 9 out of 10 character differs between species 1 and 3, 90%, and that gives us this distance. And based on this table of pairwise distances, we can cluster the species into groups, and that's called a heap map, because if distances are small, that gives us light boxes. We see that all these 4 distances are small, and so is it up here, whereas the small circles are dark for pairs of species from the same from different groups when the distance is large. So in this case, this suggests that these 4 species are divided into 2 groups. 1 and 2 form their own group, and species 3 and 4 form their own group. So let me give an example of a heat map with a real and much larger data set. And it consists of 33 animal groups or species, and we have, instead of 10, 20,000 characters, which are really proteins. And here we have actually modern humans, Denisovans, and Neanderthals. And this heat map suggests that humans form its own group. I will return to this example later, but it's very strong evidence for humans being unique using a huge data set. So that was the first goal of baraminology, trying to identify. Yes. Yeah. It's a very large data set with 20,000 proteins. So different proteins are active in different tissues in the human body. But I will return to this. But these authors, they use sequence alignment methods to identify the proteins, and it could be all different kinds of proteins. But because different species can have the corresponding genes located on different chromosomes. So it's quite complicated to pair, to find the gene in one species that corresponds to another gene that codes for the same protein in another species. The answer is that it's a large variety of different proteins. Yeah. And then we go to the second goal of baraminology, to find the mechanisms of divergence and speciation within each kind. And as I mentioned earlier, a biblical motivation is that God created each kind to rapidly adjust to changed environmental condition. And in God's original creation, we can think of two major features. And here I have illustrated this with the created kinds. Here we have the first generation. Here we have 4 species today. So we have 3 speciation events where new species are formed. And then we need mechanisms of change in order for those speciations to happen. But we also have created diversity in the first generation. And so God created the first generation of organisms of that particular kind, like a Swiss Army knife, where including the possibilities to adjust to environmental changes in the future. And then based on that created diversity in the first generation here, this design mechanisms of change could take place and cause speciation later on in the history of this particular kind. And that implies that in many kinds we have lots of species today, and that means that the observed degree of speciation in each created kind is possible within a young Earth creationist time frame, because these created diversity and the design mechanisms change. They are very effective, so we can have speciation taking place very quickly. So contrary to what macroevolutionary biology believes, that it has to take a long time. So speciation within a kind, because it's designed, can happen very quickly. But then we also have another evolutionary non-design mechanism of change, and they are much less efficient, and these are deleterious changes. Most well known are mutations from one generation to the next, like copying errors, and we can think of them as an effect of the fall of man. So God's creation was perfect, but then we have decay in nature after the fall of man, and they can also actually, to some extent, cause speciation. We can have chromosomal rearrangements and things like that because of this genetic decay, and that can lead to speciation as well. So let's look more closely into, first of all, the created diversity. And this is really a topic of its own talk, so I will only go through this quickly. So I will talk about created diversity as the first example of created heterozygosity as the first example of created diversity. Then there is something called variation-inducing genetic elements or transposable elements, and introduced by Barbara McClintock. But creationists Todd Wood and Peter Borger, they saw that they were important also as a created diversity factor. We also have epigenetic codes and structures that are important for epigenetic changes to take place later on. Tommy Alto is an expert on this. He gave a talk on this yesterday. But we also have redundancy. We know that some plants, for instance, they have 2 systems of photosynthesis that work for different climates, and they can be switched on and off. So it's like God's foresight in the first generation. And then we have engineering principles. Randy Guyutsa has written about this. We have, like in the cells, there are sensors, and based on these signal molecules from the sensors, we have logical circles, and then we have actuators that make these changes happen, often through epigenetic mechanisms. But all these engineering principles, they were part of the first generation. So these are different examples of God putting variation into the first generation in order to later on have quick speciation. So let us look slightly more into created heterozygosity. So we know that humans, for instance, have 22 pairs of non-sex chromosomes. So here we have Adam had 2 copies of the first pair, Adam had 2 copies of chromosome 1, and so on, until 2 copies of chromosome 2, the same for Eve. And we know that each DNA molecule is a double-stranded string of nucleotides. So we have a number of base pairs. So suppose at a particular position, Adam had, for one of his copies of chromosome 1, he had a C along the coding strand, whereas he had a T along the coding strand of that position for the other chromosome. Then that is called a single nucleotide polymorphisms. And God could have put lots of such single nucleotide polymorphism variation into Adam and Eve in the first generation. And this is not like mutation. So it is neutral variation that leads to diversification among the offspring later on. Then we have the second principle, variation inducing genetic elements or transposable elements. This is one example of such an element, a transposon. So this is part of DNA from one chromosome that is transferred to another part of the chromosome. And that was the possibility of this was a part of the first generation in order to later on in the history of the kind make it possible for speciation to take place. And we have epigenetic structures, and these are histone proteins, and the DNA, the DNA molecules are wound around a collection of those histone molecules, and then we have codes like molecules attaching to those histones, and that determine whether the DNA string will open up at that place, and then those genes there will be expressed to proteins. And the codes for that to happen, they needed to be present in the first generation. And that, as I mentioned, then we also have redundancy and engineering principles for environmental tracking as 2 other examples of created diversity in the first generation. So that was created diversity. Let's now look at the mechanisms of change that lead to speciation. And they can be either designed or non-designed. We have both these types. So we have, for instance, we can have a genetic drift and founder effect. So when a new population is founded at an island, then the descendants, their genetic makeup will be influenced by the particular makeup of the founders. And that can sometimes lead to speciation. And so that's a purely genetic effect. So it could be designed, but we could also have non-designed. These founders could have some inheritable diseases that are spread to the offspring. And mutations, we usually think of them as non-design, they are copying errors. But there could be some design mutations. For instance, in the human, in the immune system, we need lots of genetic diversity. And some of those mutations can be designed, actually. Then we have crossovers, and that is for 2-sex organism, we get half of our DNA from the mother and half of from the father, and crossovers are points on switches between grandmaternal and grandpaternal inheritance. And that's part of God's design, because he wants more variation in the offspring. Then we have natural selection, and so it's we think often of nature doing the selection, but also individuals can sometimes decide to move to a new place and start a new group, like having a founder effect. And then maybe the most important of all mechanisms of change, the epigenetic changes, because they are like switches above the DNA level that can turn on and turn off genes, and epigenetic changes can happen very quickly. There are many types of epigenetic changes, and they are usually reversible as well, whereas some of these genetic changes are not. And then we have also developmental plasticity, changes during embryological development. Usually, it's very harmful to have mutations during embryological development, and the fetus will not survive. But the researchers have found that there is still some plasticity in embryological variation, and some creationists have suggested that that could also lead to some speciation. So that was the second goal of baraminology, to find the mechanism of change within each group. Let's now turn to the third group to find the ancestral history of each kind. So suppose that a kind has been identified, and that's the first goal of baraminology, and then we want to find the ancestral history. And that includes biogeography, often due to environmental changes. So creationists often believe that plate tectonics took place during the flood. And of course, that caused continental drift, and that also influenced the history of that particular kind, geographic spread. We also have animals rafting on debris, like pieces of wood and things like that. They could also explain geographic spread, but also climate variations, like the Ice Age a couple of hundred years after the flood, also influence the geographic history of each kind. But we can also look at this genetically to find the history of each kind, and then we can use mechanisms of change, in particular in DNA, to trace the history of each kind. So for two-sex species, and like for humans, we have only men having Y chromosomes, and so this is a Y chromosome tree for all men alive today. So the men alive today correspond to the tips of the tree, and then somewhere here in the middle we have the most recent common ancestor of all men. And that gives us, we could do similarly for other kinds and build their history for kinds with men and women. We could build Y chromosome trees. And it's also the case that mitochondria, these are the power or energy stations of the cells. They have their own DNA. It's only inherited from the mother. So we can similarly build, based on mitochondrial DNA, we can build female trees. So the tips are all women alive today, and somewhere in the middle we find the most recent common ancestor. And amazingly, it turns out that these trees, the time scale of these trees, both for the male and female trees, are consistent with the young Earth creationist time scale, less than 10,000 years ago. How long does it take to reach the most recent common ancestor? Due to work by Nathaniel Jensen, 2013, and Nathaniel Jensen and Jason Lyle from 2016. So let's look more closely into the human Y chromosomes, or mitochondrial DNA tree. So, as I said, for the human Y chromosome tree, the male tree, the tips are men or women for the mitochondrial DNA tree alive today. And then we have some key questions. So data only gives an unrooted tree. We don't know where along this tree the most recent common ancestor lived. We would like to find the root of the tree. And another key question is, how fast do the branches coalesce, go together? Because that is related to when did the most recent common ancestor live. So let's look more closely into the age of the most recent common ancestor of the tree. And that is related to mutations. And let's look here at the branches of a Y chromosome tree, a male tree. They differ because of mutations. When a father passes on Y chromosome to his son, or the mother mitochondrial DNA to her daughter. And branches that differ by fewer mutations merge more quickly. And how quickly depends on the mutation rate and the generation time. So here we have an example. So here are 5 men alive today. 23 mutations are unique for this man, 28 for this man. 18 are located in their 2 chromosomes, but not in the others. And continuing in this way, you can build this tree. That's the principle for how to do it. Let's assume now that the root of the tree is located here. So we have 25 mutations to the left of this arrow and 61 to the right. And that makes it possible to build a rooted tree, actually, because then we start from here, and then we have 25 mutations here because before we have the first split here, and so on, and then we get an unrooted tree once we find the root of the unrooted tree. So that's the principle. But we need, when did this founder live? Then we need to know the mutation rate. And the mutation rate is the number of mutations per generation. And the faster it is, the more recently the most recent common ancestor lived. And now it's only the last couple of decades that the mutation rates has been estimated with accuracy using the so-called pedigree methods. And pedigree methods is basically comparing, in this case, the Y chromosome between father and son, and count the number of differences. And having a large number of father-son pairs, you can estimate the fraction of your mutation rates per generation. And this is a key point. It's much higher than previously believed, when the mutation rate could not be measured in this way, by at least an order of magnitude higher. And that implies that the age of the most recent common ancestor is of the order 5,000 to 10,000 years for a wide variety of species. And it was believed to be more than 100,000 years earlier. So let's now study humans in more detail. And I will argue that humans are unique, its own created kind, in 3 ways. Firstly, we will use statistical baraminology to argue that humans are genetically different from great apes and other species. And going back to this study I mentioned earlier, secondly, I will use non-sex chromosome data to argue that humans' genetic variations is consistent with ancestry from a first couple, Adam and Eve. And thirdly, we will use the female tree from mitochondrial DNA and the male tree from Y chromosome data to build humans' female and male ancestral tree with a time frame that is consistent with young Earth creationism. So let's start with this statistical baraminology study that I mentioned about, the paper by Leitner and Serrati from 2019. And it includes 4 cat animals, mice and rat animals, 2 of them, 18 monkeys and apes, including some great apes. And then we have modern humans, 3 groups, modern humans, Neanderthals and Denisovans. Chicken-like birds, 4 species, goose, ducks and swan animals, 2 species, and an African frog. And as I mentioned, this is a huge data set with 20,000 proteins, and for each species group, we mark whether that each particular protein is absent or present. And here, and then I go back to what I mentioned earlier. These proteins can be located on different chromosomes for different species. So you need so-called sequence alignment method, where you need to align the same genes or corresponding genes from different species that might be located on different chromosomes in order to find the corresponding genes and hence the corresponding proteins that these genes code for. And the result is amazing. Here we have this heat map. And it reveals that humans are clearly unique, because here we have humans form its own cluster according to this heat map. And the 4 great apes including gorillas and chimpanzees. They are more closely related to other species in this study than to humans. So it's quite very, because it's a huge data set, it strongly indicates that humans are unique. So let's now look at the second line of evidence to support that human uniqueness. And as I mentioned, we humans have 22 non-sex chromosomes, and the total length of them is about 3 billion nucleotides. And I mentioned about biallelic SNPs. These are positions where there are variations among humans. So suppose that we have one particular position here, a base pair, where in 40% of the human population we have C along the coding strand, so a base pair CG, and in 60% we have a T along the coding strand. So that's the SNP, that particular position. And then the minor allele frequency of a biallelic SNP is the frequency of the less common allele. So if C occurs in 40% of all humans at this position and T in 60%, then the minor allele frequency is 40%. So generally it's between 0 and 50%. And there are about 10 million SNPs with a minor allele frequency of at least 1%. And that's 0.3% of the genome. And the interesting thing now is that we can build an allele frequency spectrum, and that's an histogram of all of the minor allele frequency of all 10 million SNPs. So here we have all the 10 million SNPs, and how many are there with this particular minor allele frequency, and this particular, and so on. And the shape of the minor allele, of the allele frequency spectrum gives information about the history of humanity. So usually an old population has a higher fraction of SNPs with a small minor allele frequency, whereas for a young population it tends to be more flat. And for a creationist model, you can think of many of the alleles with a large minor allele frequency. They correspond to this created heterozygosity I mentioned about, God putting variation into the chromosomes of Adam and Eve. Whereas these SNPs with a small minor allele frequency, they correspond to more recent mutations. Whereas in a tree of life model, you don't have the concept of created heterozygosity. You only have mutations. So then these SNPs with a high minor allele frequency correspond to old mutations, and these ones with a small minor allele frequency correspond to recent mutations. And I published a paper with Anne Gauger 6 years ago, and there is another paper by John Sanford et al. where we fitted allele frequency spectra using chromosome data from chromosome 22 to a creationist model and an evolutionary tree of life model. And we had at least as good as fit for a creation model as for the tree of life model. But I think we have even stronger case for the creationist models using mitochondrial DNA or Y chromosome DNA. These female trees and male trees I mentioned about earlier. And I will tell you a little about the history of building up those trees. So in the 1980s, the first estimates of the age of the most recent common ancestor of the female tree was dated to between 100 and 200,000 years. The mitochondrial Eve, you have probably heard that name. And they depended on evolutionary assumptions, because basically you compared DNA of mitochondrial DNA among humans and then with chimps, and then assuming that the split was 6 million years ago, they estimated that the time of the most recent common ancestor for humans was 150,000 years ago. So this was based on evolutionary assumptions, so kind of circular reasoning. But in the mid-1990s, the first estimates of human mutation rates by the pedigree methods, I illustrated this earlier for Y chromosomes. It's the same principle for mitochondrial DNA. It's circular. And then we basically compare a number of mother-daughter pairs and check what is the fraction, average number of mutations per generation. And the first estimates of mutation rates by pedigree method, they gave conflicting results, but generally the age of the most recent common ancestor was found to be at least 1 order of magnitude smaller, because the mutation rate was 1 order of magnitude larger, and hence the age of the most recent common ancestor at least an order of magnitude smaller. And then in 2013 and 16, Nathaniel Jensen, he pooled all these studies and obtained a most recent common ancestor less than 10,000 years ago, consistent with Eve. He also identified 3 major branches from the root, and that could correspond to the 3 daughters-in-law of Noah. Because if you take the female line, it doesn't pass the wife of Noah, because then it goes through the sons, and this is a female tree. So that is consistent with it. It's quite amazing. And then if we look at the Y chromosome human tree, it's a similar development. In the 1990s, we had the first estimates of the age of the most recent common ancestor, and that and those gave numbers several 100,000 years based on low-quality DNA data. Then in 2019, Nathaniel Jensen and Holland, they pooled several studies with high-quality DNA, and they estimated the mutation rate with, again, the pedigree method, which is much more reliable, really more empirical, comparing DNA of fathers and sons. That gave a most recent common ancestor living about 4,500 years ago. And according to the Bible, all men are descendants of Noah. That is the most recent common ancestor, not Adam. And that is amazing that you get this number. And then a couple of years later, Nathaniel Jensen published the book Traced with a young Earth creationist timescale of the Y tree. So also secular scientists, they build the same unrooted tree. So everyone agrees on the unrooted Y chromosome tree. As I mentioned, the question is where to put the root and how fast do the branches coalesce. And in this book, he managed not only to identify the root 4,500 years ago, but also 3 branches merging from this root that then correspond to the 3 sons of Noah. And also later on, the early branches of the tree, they could correspond to descendants of Noah a couple of generations later on that we read in Genesis chapter 10. That's then are sort of founding different subpopulations over the world. So let me end with some summary and conclusions. So the study of created kinds, it involves 3 major goals. Firstly, to find all created kinds. Secondly, to find mechanisms of divergence and speciation within each kind. And thirdly, to find the ancestral tree history of each kind. And this is really an intensive field of research. And as I have hinted at during this talk, it includes lots of new molecular data, genome-wide data, ancient DNA from species not alive today. We know that we can sequence DNA from Neanderthals, for instance. And we also have many sophisticated tools. I mentioned statistical baraminology, and they were based on sequence alignment methods to identify proteins from different species that could be located on different chromosomes. We also need tree building algorithms in order to build this tree and so on. And the results, they clearly show that the data group species into kinds. There still remains a lot of work to exactly identify all biblical kinds, but generally data really show a clear grouping of species into different kinds. And we have designed mechanisms of change that allow for quick speciation within kinds, which is consistent with the young Earth creationist timescale. Speciation can happen very quickly, and about the history of each kind. We can use mitochondrial DNA for female trees or Y chromosome DNA for male trees for species with men and women or two-sex species, and find that those kinds have a recent ancestry, also in agreement with the young Earth creationist timescales. And then we looked, at the end of the talk, I looked at humans, and the conclusion is that humans are unique using genetic analysis. And that also confirms the Bible and also human history. And my prediction for the future is that future research, based on even more data, we have large consortia like the Biogenome Project, where the goal is to sequence basically all species, to sequence their DNA, and then we get even more data to sort of identify the biblical kinds. That this analysis of this new data will continue to strengthen the bushes of life model and of human uniqueness. And hopefully more people will realize how reliable the Bible is, because this is really described in the first chapters of the Bible about the biblical kinds, and that in turn will lead them to give their lives to Jesus. And let me end with a quote from the book of Romans chapter 1, verses 19 and 20. Then the Apostle Paul writes, what may be known about God is plain to them, because God has made it plain to them. For since the creation of the world, God's invisible qualities, his internal power and divine nature, has been clearly seen, being understood from what he has made, so that people are without excuse. So we can see God behind what he has made, and that also includes that he created animals and plants after their kind. And we can see this from his creation, and even more clearly when we analyze data, recent data that scientists bring forward to us. And thanks for your attention.